Before we do, are there any announcements for the good of the group? All right, I'm glad to see a big crowd here. We doubled our attendance from last week. So this is great. If we keep doing that, we'll have to get another room by the end of this month. All right, today's speaker is Tomas Hasing, and he's originally from Ecuador, where he was born and raised. He did his undergraduate work in Honduras, and then his bachelor's at Clemson, his master's at Clemson in applied economics and statistics. And now he's working at Virginia Tech, and he's from the department originally known as horticulture. So today's seminar is on a horticultural topic, so I think it'll be interesting to all of us. And I'll turn it over to Tomas.
Thank you, John. First of all, I would like to thank everybody for coming to my seminar. I know that my research might not be in line with what you guys are used to in PPWS, but I hope you guys enjoy it. So today I'm going to talk about the domestication and the genomics actually behind the domestication of Sinningia speciosa, which, I don't know, without counting people that I work with in my lab, who knows about this plant? Okay, so it's very popular. I didn't expect that. So the common name is actually known as Gloxinia, Floris Gloxinia, and that's actually because originally it was placed in the wrong genus, and it was later changed into the Sinningia genus. So plant domestication, simply put, is the selection process to adapt wild species for human uses. And here are just a few examples of the phenotypic changes that we have imposed in major staple food crops. Okay. And these are just some of the changes. Of course, we also have changes in plant structure, climate, things like that. But just as an overview. Also, I want to put you in context of plant domestication a little bit. The numbers that you see on top are actually in thousands, so those are thousands of years. And it's generally accepted that plant domestication actually started between 10 and 12,000 years ago, with wheat and barley actually among the oldest crops around. In this graph, of course, was made by me. It was made by Lars in 2014. But this graph is actually huge. I just chop it out and put some voices in here just to show you some of the most important crops. You see that we also have among the oldest ones corn, which was domesticated about 9,000 years ago, and rice and sunflower about 5,000 years ago. So I give you a little bit of the time frame on plant domestication. So it makes complete sense that actually scientists have put a lot of effort in studying the plant domestication of very important staple food crops like rice and corn and wheat and such. And actually ornamental crops, in general horticultural crops have been left around with not much studies until recently. So recently more studies are actually popping up, and I think that's great. We're learning more about plant domestications in different traditional crops. But horticultural crops is still underrepresented, especially if we talk about ornamental crops. We practically know nothing about the domestication of ornamental crops, except for a few cases. For example, in roses, where the domestication of roses has been described indeed. Now, knowing about every single crop, every crop that actually diverges from traditional staple crop, even ornamental crops, and algae that are being grown, and other horticultural crops is great because it gives us a broad perspective on plant domestication to kind of pick on general trends, modern trends that are actually happening because the world is different than it was 12,000 years ago. So why did we choose Sinningia speciosa? We actually think that it could be an excellent model for crop domestication. I'm not going to bore you, I'm not going to read through all this list, you can do it yourself, but I'm going to point out the two major points that I think are really important. The first one is that it's been cultivated only for 200 years. And if you put that into context, the time frame that I showed you before, that's barely a tiny amount of time compared to wheat or corn, all those crops. And the second most important one is because during that very short period of time, we have created a huge amount of phenotypic diversity. So you might be thinking, wait, what about roses and tulips and amaryllis? And all those crops also have a lot of phenotypic diversity, and that's true. But for example, in roses started cultivation about 5,000 years ago, so it's been around for a while. And moreover, it's estimated that there's about 8 to 20 different species in the background of domesticated modern roses. So that interspecific hybridization might have boosted the phenotypic diversity that we actually see in all those crops. A little bit about the history of Sinningia speciosa. The species is actually native to the southern coastal forest biome of Brazil. But in 1815, the plant was actually introduced into England, and it actually became really, really popular as an ornamental houseplant. Within a couple of years, it got really popular in the country, and then soon after that, it actually expanded to other portions of the world. And it reached the US possibly as soon as 1819, so that's really, really fast. But one thing that I want you to focus on is actually these two different flowers, because these are the wild types found in Brazil. And in 1845, actually, this phenotype appeared in England. And this mutation is actually really important because this shape of flower is actually what's being fixed among all the cultivated material, most of the cultivated material that we see nowadays. So keep that in the back of your head. So the objectives, at least for this presentation, is to describe the genomic changes associated with the rapid increase in phenotypic diversity across cultivated material of Sinningia speciosa. And to do that, we're going to be looking at potential hybridization events, genetic bottlenecks. We're going to try to identify potential founders of the domesticated types, and identify some loci associated with domesticated traits. Now, I'm going to tell you a little bit about the plant material that I use for this study. Right here, over here, is the tribe Sinningia, and I don't want you to actually read through all these little names over there. What I want you to pay close attention that there's actually three very big groups in the tribe: Dircaea, Corytonoma, and Sinningia. This is a very, very diverse group of plants with very different growth habits, and as you can see in this picture, very different pollination syndromes. And what's more interesting about this group is that interspecific hybridization within the tribe, even across clades, is very common, and I'm going to show you some examples later on. So here is our favorite species, Sinningia speciosa, right there. And therefore, that's the species that we're interested in. Well, in total, we actually genotyped 115 individuals. Out of those, 58 were Sinningia speciosa, and you can see the breakage over here. But then, since we were interested in actually looking for potential hybridization events, we also sampled different species across the whole tribe. And you can see some of them pointed over there. So that accounted for about 57 other individuals, 32 species, and 9 interspecific hybrids. This is just the location of a few of the populations that are included in this study. I actually didn't go to Brazil to sample all these plants. I would like to do that. But this are actually samples. This is plant material that has been brought to the US during a period of a few years, and they have actually been maintained in the US. But that gives you a little bit an idea about the range of the geographical region. So we took the plant material, we extracted DNA, we prepared libraries, and we send all those libraries for sequencing. I'm not going to bore you with the pipeline that I used to call variants and whatnot. If you have any questions about it, just let me know. We can discuss it. So the first thing I did is actually to create a principal component analysis on genetic distances using those SNPs. And this figure over here refers to the entire tribes in India. Okay, and we managed to actually keep almost 10,000 non-missing SNPs. So I think that's an amazing number of SNPs, of single nucleotide polymorphic markers. If you don't know what I mean, considering that this is a very diverse group of individuals across the entire tribe. Another thing to pay attention is that between the two first principal components, we explained about 95% of the variation observed. Now, if you remember actually the three clades that we saw in the tree that I showed you before, you see that they separated very nicely this principal component analysis, so that was very good news. The second thing I want you to notice is that Sinningia speciosa clusters right here. They're all tied together. These are cultivated types, these are wild types, and some F1 process that we created to test. Another important thing to notice in this graph is that actually interspecific crosses always perfectly fall in between the parents. In this case, you can see YMA falls in between those parents. Mount Magazine falls in between those parents. And you can actually tell that these are interclades, hybrids. And here, for example, this unnamed hybrid also falls between two parents, Sinningia speciosa and Sinningia helera. So that's very important to notice, because given that Sinningia speciosa is clustering all together the wild types and cultivated types, there's no actual evidence of interspecific hybridization pulling cultivated materials away from the wild types. So we are discarding actually the idea that any of these phenotypic variations has been introduced through interspecific hybridization. The next thing I did was actually, I wanted to gain a little bit of knowledge just within Sinningia speciosa, of course, because that's what we're studying. So I created another principal component analysis, just taking into account individuals from Sinningia speciosa, and you can see that because we have less individuals and they're closer related across each other, we actually managed to keep way more non-missing SNPs, about 25,000. So I think that's a really good number of SNPs. And you can see, although that between the two principal components, we only reach about less than 70% of the variation, which I think is still pretty good given the amount of SNPs that we have. Now this group over here is the domesticated types. This group over here we call actually semi-domesticated one. Why? Because it's a group of cultivars that are actually very old. And even though they're cultivated, they are very wildish looking. If you look at any of these, it will be hard to differentiate it from any of the wild types. But they're actually good. And over here we have actually the range of the wild types. And just by looking into the graph gives you the idea that we have actually lost a significant amount of genetic variation, and we'll get to that later on. Just following the same trend as the interspecific hybrids, when you look at the intraspecific hybrid, you see that we observe the same trend, they fall right in the middle of parents. But the most important thing to notice here is that we only managed to find two wild types that cluster together with the cultivated types. And these are Avenida Niemeyer and sadly an unknown genotype. We don't actually know but we know a lot about this individual here. So from here we can kind of think, okay, maybe these are very important founders for the material. And when I mean that, I don't say these particular individuals, but the populations where these individuals came from. So just to give you a little bit of an idea about the phenotypic diversity across wild types, I put some pictures here across the graph, and also of the cultivated material over here. And I know that you cannot see those very well, so I made them big here, and you can appreciate phenotypic diversity of cultivated material compared to the wild types. You can see that we have double corollas, different colors, different patterns of colors, different shapes. We would have whites. So much diversity compared to the purplish looking clouds. Now we did a population structure analysis, and if you're not familiar with this type of analysis, what we have here in each line is actually one individual. I just happen to organize them according to the groups of cultivated wild types, the semi-domesticated ones that I talked about, and some test crosses that we made F1s. So the downside about this type of analysis is that you actually have to estimate the number of clusters of groups that you want to input into a mixture, and you have to estimate that. And our estimates were not very clear. They kind of range between 2, 3, and 4. So instead of picking just one at random, I'm going to show you the results, assuming 2 populations. They're not really population, more like grouping. Some people call it ancestral populations, but that's a little bit of controversy about that. 2 groupings, 3 groupings, and 4 groupings. So depending on the height of the bars, different colors, that means that particular individual is actually a mix between the 2 groups. So at population number 2, K equals 2, you see that cultivated types and wild types actually separate perfectly. And except for Avenida Niemeyer and that unknown wild type over there. So that actually supports what we saw with principal component analysis, that these are potentially founders of the cultivated material. When we go to K equals 3, the only thing that happens, the wild types actually start to break apart. Now we have 2 main groups here, and actually Avenida Niemeyer and the other one, he being associated with domesticated types. When we go to K equals 4, actually the domesticated types start to break apart. However, if you see the ones on the right, these are actually the oldest cultivars, and they remain associated to the very old semi-domesticated types, which are still associated with Avenida Niemeyer and the wood type. So we believe that because these are modern cultivars, have gone through additional cycles of breeding, they have started to differentiate themselves from the other types, and that's why they're clustering separately from these guys over here. Now, to confirm this, we actually also fitted another model. This is a fine structure. This is more of a Bayesian model based on actually haplotypes. But I'm not going to go into too many details here, but you see the exact same thing, wild types over here, domesticated over here, and Avenida Niemeyer and the other one actually falling within domesticated types. One thing to notice here that I think is very, very useful is that you see Avenida Niemeyer and the unknown actually fell within the same population. So even though it's an unknown individual, it's very likely that actually came from the same population that Avenida Niemeyer came from. And just by looking at how these wild types were separated over here, I'm pretty confident in the results, because the populations separated on these wild types they separate perfectly according to the location. So it's very likely that these 2 individuals came from the same population. Now I talked about diversity before, and we just need to put a number behind all those figures that I showed you before. So I estimated popular estimates of genetic diversity using data Watterson and high coefficients. And you don't need to know what those numbers mean, but what you need to see is that actually the change between wild and domesticated types. You can see that there's also about a reduction between 40 to 60% on genetic diversity, at least in the coefficients. So yes, that's a pretty significant bottleneck reduction of genetic diversity, but what impressed me the most is that, for example, through domestication, at least in the samples that we have right here, we have fixed about 72% of the SNPs that are present in the wild types. So I think that's a very impressive number. And that number that goes all the way up to 86% if we only consider minor alleles with frequencies less than 5%. Now comparing that with something like rice, for example, where 82% of SNPs, this Rufi Pogon, which is the wild relative of cultivated rice, also segregate in Oryza sativa. So the relationship is kind of inverse, very, very strong bottleneck in Sinningia speciosa compared of what we see in rice. Now I also we created a F2 mapping population. We used Empress, which is a cultivated type obviously, and Bucius. We created an F1 and went all the way to F2 and we created a population of 160 individuals. We phenotype and genotype them using GBS, just like the other group of plants. Now a little bit about the summary statistics is that we got these frequencies of different colors, purple and red, which segregate 3 to 1, which is a perfect Mendelian segregation. So okay, this might be a single gene. Then we wanted to look at the phenotypic of the flower shape, the zygomorphic versus actinomorphic flowers. That was a little tricky because if you see down here, it's not like a black and white type of scenario. There's stuff in between. So what we did is we actually measured the angle of the flower as a proxy to flower shape. And what we got is a distribution of this shape, bimodal distribution, with the pines falling into extremes. But if we actually look at the number of individuals on these peaks, we actually found a 3 to 1 segregation, so we say, okay, maybe it's also a single gene. And that was confirmed through PTL analysis. We got actually very nice results, very strong PTLs and different chromosomes for both traits actually. When you look at flower color, you see the PTL is kind of on the edge of the chromosome, so I think that we don't actually have the scaffold that contains the gene that we're looking for, so I haven't been able to pin down particular candidate genes for that trait. However, for flower symmetry, you see that the peak falls in the middle of the chromosome, and we actually find a very strong candidate gene for that trait. So this is my very fake representation of cycloidea gene. Cycloidea gene is being found responsible in other plants like snapdragon to be responsible for the asymmetric shape of the flower. And this gene just happens to be right in the middle of our PTL. And when we start aligning plants that are zygomorphic, we see that they all align perfectly. We usually find some SNPs here and there, but they're mostly synonymous SNPs, or they fall in the introns or these type of areas. But when we look at the actinomorphic flowers, we find this deletion. This is actually 2 small deletions right next to each other, 7 base pair next to a 3 base pair. And in the middle, there's like a 2 base pair that is not missing. But these segregated perfectly with the phenotypes that we observed for this trait. So we're fairly confident that this was the trait that the gene that we're going for. And then after 6 months, a Chinese group published a paper describing this exact gene in Sinningia they did transformations, and they confirmed that it's exactly the gene that we just found just a few months ago. So this is a little bit of a sad story, but this is my cartoonish representation. If you are interested in how actually data looks like, this is a little bit messy, but the red ones are actually the domesticated types, and you see here the deletion that I'm talking about. And we have way more individuals than these. This is just a few ones that I've just shown you. The most important thing to notice is that this is the exact same mutation that is creating this shape. So it's very unlikely that you're going to have the same mutation twice in the same gene. So this is very likely to happen only once, and it was in 1985 in England. And from that single plant, we have managed to disperse that mutation into every single cultivated plant out there now. So it's been a very strong problem, genetic sweep, as we domesticate this plant. So conclusions. We have all this huge amount of phenotypic diversity. How did we come up with it? It was clearly not hybridization events, interspecific hybridization events. But not only that, but we have managed to create such amount of phenotypic diversity with individuals that came from only a single population from Brazil. So very reduced genetic diversity. Not only that, but we put all those plants through this very intensive genetic sweep around the cyclopedia gene. And still we managed to come up with this huge amount of phenotypic diversity. How did we do that? I don't know, but I think it's very helpful to know that diversity appears to be relying on traits that are controlled by single genes. I know I'm only showing 2 traits over here, but we know, I think actually the double corolla trait is very likely to be controlled by also a single gene. We have also preliminary data on segregation studies suggesting that that's the case. Now, from a more applied point of view, like talking about breeding, for example, we have identified what populations that actually can be used to increase the genetic diversity on our breeding population. So, very useful information. And also, interspecific hybridization could potentially be used to produce newer phenotypes among cultivated material. And with that, I would like to thank members of my committee, all the people who has helped me doing all this research. Jim Roberts was a crucial person in providing some plant material. He's actually a member of the Sinningia Society. Also, Dave Saving provided a lot of material. He's also a member of the Sinningia Society. And of course, the previously known as Department of Horticulture funded this research, and the Sinningia Society also provided a little bit of the funding. With that, I will be happy to take any questions.
How'd you select the parents for your master population? What were the choice?
Let me say first that I didn't select the parents. The population, the F1 was actually by Dave Saving, who is an expert guy in Sinningia. He loves these plants. He actually works in University of Kentucky, and he works on tobacco, but Sinningia is his passion. He's probably the guy who knows about Sinningia the most in the world. So he chose these parents mainly just because it was a cultivated type and it's a wild type, segregated in color. He was actually very interested in identifying the gene responsible for color, which I haven't been able to find yet, but hopefully we'll find it later, because we're trying to assemble a better genome, putting all the scaffolds into chromosomes, and when we do that, hopefully we're going to be able to find the gene.
So are there other traits that you're following in that population?
Right now we have those. I have actually measured a bunch of other traits, but I haven't actually had the time to put it all together and do the extra analysis. Yeah, but sadly, double corolla, double against single corolla is a very important trait, but it's not segregating obviously in that population. So probably that's something that we're not going to be able to identify in this research.
So I have a question on your structure analysis. Initially, you showed results from 3 different K values, 2 through 4. Was one of those had an optimum log likelihood score, or why did you show all 3 as opposed to?
Yeah, I didn't actually use structure. I used a mixture, which is very similar. Kind of uses the same algorithm, but the optimization process is different. But the thing is that you have to optimize the function, but there was a value, and the value was 2, 3, and 4. There was not a clear difference between which one is the best, and that's why I decided to put both of them, because it was just a value that came 2, 3, 4, and then it went out, and there was not a clear winner.
So you suggested that selection of these genes for diversity, at least in some of these floral traits, are single genes. You sort of intimated that you were surprised by that. But they're typically single genes that are transcription factors. Right? So I'm wondering, do you think that has some role in it, and do you think that this is maybe a biased analysis because you're focused on things that have to get fixed really quickly?
Oh yeah, definitely it's biased. For example, things that I wouldn't expect to be controlled by a single gene and flower size. There might be some effect by cyclo-E gene on affecting flower size, of course, but aside that, I think that it's probably controlled by a bunch of different genes. Another trait, for example, might be the pattern, color pattern on the flower. Seeing reports over there that could be affected by transposomes, or also by microRNAs, and that can control that. So yeah, we're biased, of course, but we are kind of targeting the most evident traits that you can see that are affecting ornamental traits. Of course, we're not focusing on plant diseases or anything like that. But yeah.
All right, any more questions? Okay, let's start this week.